Right now, something inside you is under attack. You cannot feel it. You cannot see it. But in the time it takes you to read this sentence, your body has already identified the threat, assembled a response, and sent the first wave of defenders. All of that happened without a single conscious thought from you. This has been going on your entire life, every hour of every day, without rest, without pause, without ever asking your permission. And it started long before you were born, long before humans existed, long before animals walked the earth. The system protecting you right now has roots 4 billion years old. It was forged in darkness, in a world so alien we barely have words for it. And it has been fighting, failing, adapting, and remembering ever since. This is the story of that system, how it was built, how it learned, how it has been pushed to its absolute limits, and how, for the first time in 4 billion years, we are beginning to rewrite it. Picture the ocean 3.5 billion years ago. There is no color to it, no coral, no fish, no light filtering down through clear water. The sky above is a haze of carbon dioxide and methane. And the sun, younger and dimmer than the one we know, offers little warmth. The world is almost completely silent. But the water is not empty, invisible to any eye that does not yet exist. The ocean teems with life, microscopic, single-celled life, bacteria drifting in the dark, pulling nutrients from the water, dividing, surviving. They have been doing this for hundreds of millions of years. They are, by any honest measure, the most successful living things this planet has ever produced. And they are already at war. Not a war with armies or strategies or anything resembling a plan. A war fought at the scale of molecules. A war where the battlefield is the interior of a single cell, and the weapons are strands of genetic code. A war that most people have never heard of, and yet one that shaped everything that came after, including you. Among the oldest enemies life ever faced were viruses and the other pieces of invading genetic code that drifted through that ancient ocean alongside them. Calling a virus an enemy requires a small clarification. A virus is not alive in the way a bacterium is alive. It does not eat, breathe, or grow on its own. It is, at its core, a set of genetic instructions wrapped in a protein shell. It has no goal, no awareness, no desire. It simply drifts. And when it collides with a living cell, something remarkable and terrible happens. The virus injects its genetic instructions into the cell, hijacks the cell's own machinery, and forces that machinery to produce thousands of copies of the virus. When enough copies have been made, the cell bursts open. The copies spill out and drift away, looking for the next cell to infect. That is all a virus does, and it has been doing it for as long as life itself has existed, in a world with no defense of any kind. That process is simply death. A cell encounters a virus. The virus takes over. The cell dies. But not every cell died. Some survived, and the ones that survived were not simply lucky. Some of them had something the others did not. A small, chemical, almost accidental way to fight back. The first defense was not elegant. It was a single molecular trick, discovered by accident across billions of bacterial generations, that turned out to matter enormously. Bacteria have DNA. Viruses also have DNA. But the DNA of a virus is different from the DNA of the cell it invades. It is foreign. It does not belong. And certain bacteria, through random mutation and the relentless pressure of natural selection, developed proteins that could recognize foreign DNA and cut it apart before it could do any damage. These proteins are called restriction enzymes. The system they belong to is called the restriction modification system, one of the earliest molecular defense systems we know of in the history of life. The way it works is brutally simple. A bacterium tags its own DNA with a specific chemical marker, the way you might stamp your own documents to prove they belong to you. When foreign DNA enters the cell, it does not carry that marker. The restriction enzymes find the unmarked DNA and destroy it. The principle it established was something that had never existed before in the history of life: the ability to tell the difference between self and not self. That distinction between what belongs inside and what does not is the foundation on which every immune system that has ever existed was built. Without it, there is no defense. The restriction modification system was a beginning, crude, limited, but a beginning. And then, hundreds of millions of years later, bacteria stumbled onto something far more remarkable. Something that looked, if you understood what you were seeing, almost like memory. There is a region found in the DNA of many bacteria called CRISPR. The letters stand for something long and technical that does not matter right now. What matters is what CRISPR does. And what it does is almost impossible to believe when you first hear it. When a bacterium survives a viral attack, it sometimes cuts out a small piece of the invader's genetic code and tucks it away inside its own DNA. A tiny genetic record of the attacker, preserved in the cell's own archives. If the same virus ever returns, the bacterium pulls out that record, compares it to the intruder, recognizes the match, and destroys the virus before it causes harm. It remembers. Not in the way you and I remember. There is no experience here. No feeling of recognition. No moment of realization. This is chemistry. This is molecular machinery operating on rules it did not choose. But the outcome is the same. An encounter with a threat leaves behind a record, and that record changes how the cell responds if the same enemy ever appears again. This is one of the earliest known forms of biological memory. Not in a brain, not in a nerve, in a strand of DNA, tucked inside a single cell, drifting in a dark and ancient ocean. Sit with that for a moment. Before there were brains, before there were nerves, before there was any living thing complex enough to learn from experience in any sense we would recognize, a single-celled organism had already found a way to remember what attacked it. CRISPR has become famous in recent years because scientists learned to borrow that molecular machinery and use it as a tool for editing DNA. But its original purpose, the one evolution built it for, was defense. It was a weapon, and for billions of years, it did its job. But CRISPR has a hard limit. It can remember a specific enemy, but only that specific enemy. If the virus changes, if it mutates enough to look different at the molecular level, the record in the archive no longer matches the intruder. The memory becomes useless. The bacterium responds as if it has never seen this threat before, because in every meaningful sense, it has not. And viruses mutate, constantly, relentlessly, with a speed that makes the word rapid feel inadequate. Every time a virus replicates inside a cell, there is a small chance the copy will be slightly different from the original. Most mutations hurt the virus, but some allow it to slip past a defense it could not get through before. The ones that slip past survive to replicate again, and again, and again. This is an arms race. It has been running for 4 billion years, and for most of that time, both sides were working with extremely limited tools. The bacteria had restriction enzymes and CRISPR. The viruses had mutation and time. Neither side could win permanently. Neither side has ever won permanently. Every defense eventually gets cracked. Every countermeasure eventually gets worked around. But here is the thing about an arms race that never ends. It does not stay still. It pushes. It demands. It rewards anything that gives one side even a temporary advantage. And over billions of years, that relentless pressure produced something no early bacterium could have predicted. It produced complexity. And then, roughly 600 million years ago, life crossed a threshold that changed everything. Not just in how it defended itself, in what it was. Single cells, which had always lived and died alone, began to stay together. They began to cooperate. They began to build something that had never existed before in 4 billion years of life on Earth. They began to build bodies. And the moment they did, the nature of the war changed completely. Here is a thought experiment. Imagine you are in charge of security for a city of 37 trillion people. Your job is to identify and remove anyone who does not belong. But you have never been given a list of citizens. No photographs, no names, no identification cards of any kind. You cannot ask anyone to prove who they are, because there is no shared language, no authority to appeal to. All you have is chemistry. Now imagine that the people who do not belong are not strangers who look obviously different. Some of them look almost exactly like your own citizens. Some of them have learned to dress like your citizens, move through your city without attracting any attention at all. And some of your actual citizens have been compromised from the inside. They are still there, still walking the streets, but they are carrying something dangerous. Your job is to find all of them, every day, every hour, for the entire life of the city. This is the problem that the first multicellular bodies had to solve. And it is, without question, the hardest problem in the history of life. When the first complex organisms began to form, hundreds of millions of years ago, they inherited the basic molecular toolkit that their single-celled ancestors had developed. They had restriction enzymes. They had primitive pattern recognition. They had crude chemical alarms that could signal damage or intrusion. These tools were good enough for a single cell living alone in the ocean. They were nowhere near good enough for a body. A body is not one life. It is a community of lives, bound together by a contract, so ancient and so deep that we almost never think about it. Every cell in your body has agreed, in the wordless way that biology makes agreements, to subordinate its individual survival to the survival of the whole. Liver cells do not try to become brain cells. Skin cells do not decide to migrate to the heart. Everyone stays in their place, does their job, and accepts a fate that no single-celled organism would ever accept, the willingness to die when the body says it is time. This is a remarkable arrangement, and it creates a problem no single cell ever had to face. If you are one cell, the question of what belongs inside you is simple. Your DNA is yours. Everything else is foreign. But if you are a body made of trillions of cells, all of them descended from a single original cell, all of them carrying essentially the same DNA, the question becomes almost impossibly complicated. Which cells are healthy, and which have been infected? Which proteins belong here, and which are being manufactured by an invading virus hiding inside a cell that looks perfectly normal from the outside? Which signals can be trusted, and which have been forged by bacteria trying to blend in? The body had to develop a way to answer these questions continuously, in real time, across trillions of cells, without any central authority to coordinate the effort and without ever stopping to rest. What it developed was a language of molecules. In complex vertebrate bodies like ours, most nucleated cells are constantly broadcasting, displaying on their outer surface a selection of protein fragments that says, in chemical terms, here is what I am, here is what I am doing, and here is whether everything is normal inside me. Other cells, and the dedicated defenders of the immune system, are constantly reading those broadcasts, constantly asking the same question: does this belong? This molecular language is the foundation of everything. Without it, no immune system at the scale of a human body would be possible. Without it, your body could not tell the difference between a healthy muscle cell and a muscle cell that has been infiltrated by a virus. Early animals did not yet have the full and sophisticated version of this system, but they faced the same fundamental problem in simpler molecular forms, and the pressure to solve it never let up. The language is not perfect. It took hundreds of millions of years to develop, and it still has gaps that can be exploited, but it works well enough, most of the time, to maintain the most complex biological community ever assembled. Your cells know what they are. They announce it constantly, and the immune system listens. But announcing identity is only half the problem. The other half is knowing what to do when the announcement does not match what is expected. The earliest multicellular organisms handled this with a standing rule: if it does not carry the right molecular signature, treat it as a threat. This rule is still active in your body today. It is one of the first things the immune system does when it encounters anything unfamiliar. Check the signature. If it matches, leave it alone. If it does not, respond. This approach works well against bacteria, which carry molecular signatures that are completely foreign to animal cells. The surfaces of most bacteria are coated in structures that simply do not exist in the animal kingdom. And the immune system learned, very early in evolutionary history, to treat those structures as universal red flags. You do not need to have met a particular species of bacteria before to recognize that it is dangerous. The red flag is enough. This recognition system, this ability to detect the general signs of microbial invasion without needing any prior experience with the specific invader, is called innate immunity. It is the body's oldest defense. Older than fish, older than worms, older than almost anything we would recognize as an animal. Traces of it can be found in sea sponges, in coral, in the most ancient animals that ever left a fossil. And it is remarkably effective. Most of the time, it stops infections before they ever become serious. A splinter drives bacteria into your skin, and within hours, before you have noticed more than a slight soreness, the innate immune system has already flooded the area with defenders, begun breaking down and consuming the bacteria, and started the process of repair. You never had to think about it. The system handled it, as it handles thousands of similar challenges every single day. But innate immunity has a weakness, a fundamental one that no amount of improvement could fully eliminate. It knows types. It does not know individuals. It can recognize bacteria in general, because bacteria in general share certain molecular signatures. But it cannot recognize a specific bacterium it has encountered before and respond differently than it would to a new one. It cannot learn from experience in the way that matters most. In a war against enemies that evolve, every time the innate immune system encounters a threat, it responds with the same tools it always uses. No memory of the last encounter, No adjustment based on what worked before. No record of the enemy left behind for future reference. The bacteria in the ocean 3.5 billion years ago had already discovered, in their primitive way, that memory matters. That survival is not just about fighting off the attack in front of you, but about leaving behind some trace that makes the next fight easier. But multicellular animals, with all their complexity, had not yet found a way to build that kind of specific, targeted, long-term memory into their immune systems. For hundreds of millions of years, they got by without it. Innate immunity was enough to keep most threats at bay most of the time. Enough to allow life to grow more complex, more diverse, more elaborate in its forms. But the threats were growing more complex and diverse too. And there was a clock running, invisible and relentless, counting down to the moment when good enough would no longer be enough. That moment was coming, and when the immune system finally answered it, the answer would be so powerful, so creative, and so strange that it still seems almost miraculous, even now that we understand how it works. 600 million years ago, something happened that had never happened before in the entire history of life on Earth. A cell divided, and the two cells that resulted did not drift apart. They stayed together. This sounds almost accidental, and in a sense, it was. There was no plan behind it, no goal, no organism that decided living in groups would be a better strategy than living alone. It was a mutation, or a series of mutations, that altered the way certain cells behaved after division. Instead of separating and going their own ways, they remained attached. They shared resources. They began, in the most primitive way imaginable, to depend on each other. What followed from that one small change was the most consequential chain of events in the history of life. More consequential, in many ways, than the origin of life itself. Because what followed from cells staying together was eventually everything with a body. Every animal that has ever crawled, swum, walked, or flown across this planet. But in the beginning, it was just a cluster of identical cells, stuck together, doing more or less the same things they had always done. The first advantage was size. A cluster of cells is harder to eat than a single cell. It can store more energy, resist more damage. Size, even at that primitive scale, was useful. But size created a problem that single cells had never faced. A single cell is in direct contact with the world around it. It feels every change in chemistry, every incoming threat. It responds immediately, with everything it has, because it is everything it has. There is no division of labor. Every cell does every job. A cluster of cells cannot work that way. The cells on the outside are in contact with the environment. The cells on the inside are not. If a threat appears, the inner cells may not detect it until it has already broken through the outer layers. The larger the cluster, the more severe these problems become. The solution that evolution found to this problem is the same solution that human societies have found to the problem of organizing large groups of people: specialization, division of labor. Some cells take on the job of interacting with the outside world. Others handle digestion. Others provide structure. And some, crucially, take on the job that will eventually become one of the most complex and sophisticated systems in all of biology. Some cells become defenders. The first immune cells were not elegant. They were not precise. They did not carry sophisticated molecular weapons or make fine-grained distinctions between different types of threats. They were, at their core, cells whose primary job was to find things that did not belong and eat them. We still have descendants of those cells in your body right now. They are called macrophages, from the Greek words for big and eater. And eating is, in fact, their primary method. A macrophage is a large, mobile cell that patrols the tissues of your body, constantly probing its environment. And when it encounters something that carries the molecular signatures of a foreign invader, it engulfs it, surrounds it completely, pulls it inside, and digests it. There is nothing subtle about this process. A macrophage does not distinguish between one type of bacteria and another. It does not remember the specific pathogen it consumed last week. It finds things that trigger its danger sensors and destroys them. The same basic approach that the very first multicellular immune cells used hundreds of millions of years ago is still running in your body today. But eating the enemy is only one part of what those early defenders needed to do. The other part was communication. A single cell responding to a threat can only do so much. What a body needs, when a threat appears, is a coordinated response. A way for the cells that detect the threat to signal the rest of the body that something is wrong. A way to call for reinforcements. To direct additional resources to the site of infection. To put the entire community on alert. The solution that evolution developed was chemical signaling. When an immune cell encounters an invader, it releases a cascade of small signaling molecules into the surrounding tissue. These molecules diffuse outward, reaching other cells, carrying a message that translates roughly as "danger here. Respond now." The cells that receive these signals change their behavior. Blood vessels nearby become more permeable, allowing more immune cells to pour out of the bloodstream and into the affected tissue. The local temperature rises, creating conditions that are harder for many pathogens to survive. Other immune cells in the vicinity shift from a resting state to an aggressive, attack-ready mode. This response is what we call inflammation. And if you have ever had an infected cut, or a bad cold, or any injury that made the surrounding area red, swollen, warm, and painful, you have felt it working. Inflammation feels like a problem. It is uncomfortable. It slows you down. Every one of those things is true. But inflammation is not a malfunction. It is a weapon. One of the oldest and most powerful weapons in the biological arsenal. The redness is blood rushing to the site, carrying reinforcements. The swelling is fluid and immune cells flooding in, quarantining the infection zone. The heat is the body deliberately raising the local temperature to create a hostile environment for pathogens. The pain is a signal to you, the conscious organism, to protect the area and give the body time to work. Every one of those things is happening on purpose. And the speed of it is remarkable. Within minutes of a significant bacterial intrusion, the chemical alarm signals are already spreading. Within an hour, the affected area is flooded with macrophages and other first responders. Within a day, in most cases, a healthy immune system has the situation under control. All of that happens before you have made a single conscious decision about how to respond to the infection. This system, this combination of mobile eating cells and chemical alarm signals and controlled local inflammation, is the core of innate immunity. It is present, in some form, in virtually every animal that has ever lived. Sea urchins have it. Horseshoe crabs have it. Fruit flies, which last shared a common ancestor with humans more than 500 million years ago, have a version of it. The basic architecture has been so useful, so fundamentally reliable, that evolution has conserved it across hundreds of millions of years of change. There is something almost humbling about that. The same basic process that fights off an infection in a fruit fly is also running in you right now. The same chemical signals, the same cellular strategies, the same ancient, blunt, effective logic. Find what does not belong. Destroy it. Call for help. But there is something else worth noting about innate immunity, something that most descriptions leave out. Even this ancient system is not entirely static. Research over the past few decades has revealed that innate immune cells, in certain circumstances, respond differently to a threat they have encountered before, not with the precision and specificity that will come later in the immune system's evolution, not with the ability to remember one specific virus or one specific strain of bacteria, but there is evidence that some innate immune cells can be primed by experience, their responses shaped by previous encounters, in ways that go beyond simple reflex. Scientists call this trained immunity. It is not the same as the immune memory that would eventually evolve in more complex animals. It cannot keep a precise portrait of a specific enemy. But it is a hint that even the oldest parts of the immune system are not entirely fixed. Even they can be shaped, in their limited way, by what they have been through. This matters because the line between the old immune system and the new one is not as sharp as it sometimes appears. The transition from innate to adaptive immunity was not a sudden replacement of one thing with another. It was a gradual evolution, building on what was already there, adding new layers of capability on top of an ancient and still functional foundation. But the new layer, when it finally arrived, was unlike anything that had existed before. The innate immune system could protect a body. What came next could do something the innate system never could. It could remember a face. Cut your finger. It does not have to be a bad cut, just enough to break the skin. A kitchen knife that slips. A piece of paper with an unexpectedly sharp edge. A thorn in the garden. The kind of small injury that happens to almost everyone and is forgotten within a few days. Now pay attention to what happens next. Within seconds, the blood begins to clot. Within minutes, the skin around the cut starts to redden. Within an hour, the area is tender, slightly swollen, warmer than the surrounding skin. If the cut is deep enough, or if bacteria have entered the wound, the soreness and swelling will build over the next day before eventually, gradually, subsiding. Most people experience this sequence as damage, as the body struggling, as something going wrong that the body is trying, imperfectly, to fix. Almost every part of that interpretation is backwards. What you are watching when you watch a small wound become red and swollen and sore is not the body failing. It is the body doing exactly what it was designed to do, with a speed and coordination that no human engineering has ever matched. Every symptom you feel is a weapon being deployed. Every signal of discomfort is evidence that an ancient and extraordinarily effective system has already responded to the intrusion and is actively working to contain it. To understand why, you need to understand what a pathogen actually faces when it enters the body through a break in the skin. The skin itself is the first defense, not just a physical barrier, though it is certainly that. A multi-layered structure tough enough that most pathogens cannot penetrate it under normal circumstances. The skin also maintains a chemical environment that most microbes find hostile, slightly acidic, populated by its own community of resident organisms that compete with invaders for space and resources, covered in enzymes that can break down bacterial cell walls. When the skin is intact, most pathogens never get past it. They simply cannot. But when the skin is broken, that barrier is gone. And in the tissue just below the surface, there are cells that have been waiting for exactly this moment. Not consciously waiting, but positioned, armed, and ready to respond the instant the molecular signals of intrusion reach them. The first responders are the macrophages, already present in the tissue, already on patrol. They detect the chemical signals released by damaged cells and by any bacteria that have entered the wound. Within minutes, they are moving toward the site, engulfing bacteria, releasing their own cascade of chemical signals to amplify the alarm. Those signals, called cytokines, do several things at once. They tell the small blood vessels in the area to dilate, bringing more blood to the region. More blood means more warmth, more redness, and more of the immune cells that circulate in the bloodstream. They make the walls of those blood vessels more permeable, opening small gaps that allow certain immune cells and proteins to pass through into the tissue. And they activate other types of immune cells that are cruising through the bloodstream, telling them to stop, to press themselves against the inner wall of a blood vessel near the infection site, and to squeeze through into the tissue to join the fight. The swelling you feel around a wound is all of that happening at once. Fluid pouring into the tissue, carrying proteins that help fight infection. Immune cells arriving in large numbers. The physical pressure of that accumulation pushing on the pain-sensing nerve endings in the skin, sending a message to your brain that registers as discomfort. Your brain's interpretation of that message: something is wrong here. Pay attention. Protect this area, is actually the point. Pain is not a failure. Pain is information. It is the body telling the conscious mind to cooperate with a process that is already well underway. And then there is the fever. If the infection is significant enough to trigger a more widespread response, the same cytokines that orchestrate the local inflammation can spill into the bloodstream and reach the brain. When they do, they affect the region of the brain that regulates body temperature. Shifting the set point upward, the body begins producing more heat. You start to feel cold because your body temperature is rising toward a new target that is higher than normal. You shiver because shivering generates heat, and your temperature climbs. A fever feels miserable, but it is, in most cases, a deliberate strategy. Many bacteria and viruses are optimized to thrive at normal human body temperature. Raising that temperature even a few degrees can slow their reproduction and reduce their ability to cause damage. At the same time, many of the proteins and cells of the immune system work more efficiently at higher temperatures. A fever is the body changing the rules of the game, creating conditions that disadvantage the pathogen and advantage the defender. This is an ancient tactic. Fish get fevers. Reptiles, which cannot generate body heat internally, will move to warmer parts of their environment when infected, deliberately raising their body temperature through behavior. The strategy of heating the body to fight infection is so old and so widespread that it appears to be a near universal feature of animal life. Now stop and look at what is actually happening in your finger. The redness is deliberate. The swelling is deliberate. The heat is deliberate. The pain is deliberate. Every symptom that makes you wince and reach for a bandage is the product of hundreds of millions of years of biological engineering, deployed automatically, in the right sequence, at the right scale, with a precision that medicine is only beginning to fully map. This is a controlled burn. The body heals by first making things worse. It floods the area with destructive chemicals that kill bacteria, but also injure surrounding tissue. It generates heat that stresses the pathogen, but also stresses the host. It creates swelling that isolates the infection zone, but also causes pain and limits movement. In the short term, all of this looks like damage. In the longer term, it is what saves you. The word controlled is important, though, because the system has elaborate mechanisms to keep this response within bounds. Anti-inflammatory signals that ramp up as the infection is controlled. Regulatory cells that dampen the response once the threat has passed. Feedback loops that monitor the intensity of the reaction and adjust it in real time. When those control mechanisms work, the result is what you experience after a minor infection. A few days of soreness and swelling. Then a gradual return to normal as the inflammation resolves and the tissue repairs itself. The controls are what separate a healing wound from something far more dangerous, and they do not always hold. The same chemical cascade that saved your finger, scaled up, running out of bounds, is one of the most dangerous things the immune system is capable of producing. Not in some rare theoretical scenario, in real patients, in real hospitals, in situations that have killed millions of people throughout history. Understanding how that happens and why is where the story of immunity gets darker and more important. There is a word that has become so common in modern health conversations that most people assume they understand it: inflammation. You hear it in doctors' offices and on nutrition labels and in advertisements for supplements that promise to reduce it. You hear it described as the root cause of everything from heart disease to depression to aging itself. You hear people talk about anti-inflammatory diets, anti-inflammatory medications, anti-inflammatory lifestyles, as if inflammation were simply a malfunction that the body keeps making and that we need to keep correcting. The truth is more complicated and more interesting. Inflammation is not a malfunction. You saw that in the last chapter. Inflammation is a weapon, one of the oldest and most effective weapons in the biological arsenal, forged over hundreds of millions of years of evolutionary pressure. Without it, a small cut could kill you. Without it, every cold you ever caught would have a reasonable chance of becoming fatal. Without it, the bacteria that live on your skin and in your food and in the air you breathe would have nothing standing between them and your bloodstream. The problem with inflammation is not that it exists. The problem is what happens when it does not stop. To understand why that matters, you need to understand something about the way the innate immune system decides when to turn on and when to turn off. When a threat appears, the system activates. Cytokines flood the area. Blood vessels dilate. Immune cells pour in. All of that happens fast. Because speed is the whole point. A bacterial infection that is allowed to spread unchecked for even a few hours can become life-threatening. The innate immune system is built to respond before the threat has time to establish itself. But every activation has to end somewhere. The immune system cannot run at full power indefinitely. The collateral damage would be unsustainable. So, alongside the signals that turn inflammation on, there are signals that turn it off. Anti-inflammatory cytokines, regulatory cells that dampen the response, feedback mechanisms that monitor the level of activation and begin reducing it once the threat has been cleared. In a healthy body, responding to an ordinary infection, this system works beautifully. The activation is fast and powerful. The resolution is gradual and complete. Within days, the affected tissue is repaired and the immune system has returned to its baseline state, ready for the next challenge. But what if the trigger never goes away? What if the thing activating the immune system is not a bacterium that can be killed and cleared, but something that persists, something chronic, low level, and impossible to fully eliminate. This is the situation that produces chronic inflammation. And it is, by a wide margin, one of the most significant health problems in the modern world. When the immune system is chronically activated, even at a low level, it is constantly releasing small amounts of inflammatory cytokines, constantly running the processes that, in a short acute infection, are protective, constantly doing slight, incremental damage to the surrounding tissue over days. That damage is trivial. Over years, it accumulates. Over decades, it can fundamentally alter the structure and function of major organ systems. The connection between chronic inflammation and cardiovascular disease is one of the most well-established findings in modern medicine. The plaques that build up in arterial walls and eventually cause heart attacks are not simply accumulations of fat. They are sites of chronic immune activity, places where the immune system has been trying for years to respond to damage in the arterial wall, and has, in the process, created a wound that never fully heals. The inflammatory response meant to repair the damage instead contributes to the very instability that makes plaques dangerous. The same pattern appears in type 2 diabetes, where chronic low-grade inflammation in fat tissue contributes to insulin resistance. It appears in certain neurodegenerative diseases, where persistent immune activation in the brain may accelerate the damage to neurons. It appears in a range of cancers, where chronic inflammation creates an environment that can promote tumor growth. None of these connections mean that inflammation causes these diseases in a simple, direct way. Biology is rarely that simple, but the thread running through all of them is the same. A system designed for short, sharp, targeted responses that get stuck in a mode of chronic. Low-level activation causes a kind of damage that is fundamentally different from the acute damage of infection. Slow, cumulative, often invisible for years or decades before it becomes apparent. And then there is the other end of the spectrum, when the system does not simply persist at low level but suddenly and catastrophically overwhelms every control mechanism the body has. Picture the largest wildfire you can imagine. Now picture the fire department arriving not with a measured, targeted response, but with every piece of equipment they own, deployed all at once, with no coordination and no way to stop. The fire is still spreading. But now, the streets are also flooded. The roads are blocked, and the suppression effort itself has become a catastrophe layered on top of the original one. That is, more or less, what happens in a cytokine storm. Immune activation spirals out of control. Inflammatory signals cascade through the entire body rather than staying localized. Organs begin to fail, not because the pathogen has reached them, but because the immune response has. The body, in its effort to destroy the threat, begins destroying itself. The influenza pandemic of 1918 killed somewhere in the range of 50 million people worldwide. A figure so large, it still does not feel real when you say it out loud. More people than the entire First World War. In a matter of months. What made that pandemic strange, and what puzzled doctors at the time and for decades afterward, was who died. Normally, influenza is most dangerous to the very young and the very old. But in 1918, young adults in the prime of life died at horrifying rates. People who, by any normal measure, should have had the best chance of survival. The picture that emerged over decades of research is complex. Wartime conditions, severe crowding, malnutrition, the near-total absence of antibiotics to treat the bacterial pneumonia that followed in the wake of influenza, all played major roles in the death toll. But for some proportion of those young adults, The intense immune response that their healthy bodies could mount may have contributed to what killed them. A strong immune system, reacting powerfully to a virus it had never encountered before, flooding the lungs with inflammatory response, damaging the very tissue it was trying to protect. The weapon turned too high, became part of the wound. And this is not ancient history. Cytokine storms have been documented in severe cases of influenza, in some serious COVID-19 infections, in certain bacterial sepsis cases, and in early cancer immunotherapy trials. The same system that fights off a cold without you ever noticing is the same system that, at its most extreme, can cause multiple organ failure in a matter of days. This is the central tension of innate immunity. It is powerful because it is aggressive. It is dangerous for exactly the same reason. The line between protective inflammation and destructive inflammation is not always clear, and the body does not always stay on the right side of it. For hundreds of millions of years, this was the best the immune system could do. Fight hard, fight fast, accept the collateral damage, hope that the response clears the infection before it does too much damage to the host. It was enough, most of the time, for most animals, for most of the history of life. But the world was not standing still. Pathogens were evolving. Animals were getting larger, living longer, facing more complex and varied threats. The crude, fast, indiscriminate weapons of innate immunity were doing their job, but they were being pushed to their limits. The immune system needed something more than speed and aggression. It needed precision. It needed the ability to recognize not just the general signs of invasion, but the specific face of a specific enemy. It needed something that, when the battle was over, would leave behind more than scar tissue. It needed to remember. And in the cold, dark waters of ancient oceans, in the ancestors of the fish that would eventually give rise to every vertebrate animal that has ever lived, something new was beginning to take shape. Something that had never existed before in 4 billion years of life. Warfare and survival, the beginning of memory. 500 million years ago, a fish was swimming in a shallow sea. We do not know exactly what it looked like. The fossil record from this period is incomplete, and the specific animal in which the version of immunity we carry first appeared has not been identified and probably never will be. But we know it existed. We know it was a fish with a jaw, because the particular form of adaptive immunity that runs in your body right now, built around B cells, T cells, antibodies, and a gene shuffling process called V-D-J recombination, emerged in the jawed vertebrate lineage. And we know that whatever this animal looked like, whatever small and ordinary life it was living in that ancient sea, it carried inside its body something that had never existed before in the 4 billion year history of life on Earth. A system that could learn to recognize any enemy it had ever encountered, and remember that enemy for the rest of its life. This is adaptive immunity. And the fact that most people who know about it treat it as one item on a list of immune system components, rather than as one of the most extraordinary biological inventions in the history of evolution, says something about how difficult it is to truly absorb what it means. So let us try to absorb it. Go back to the problem that the innate immune system could not solve. The innate system could recognize types, bacteria in general, viruses in general, the general molecular signatures of things that should not be inside a body. That was enormously useful, and it still is. But it could not recognize individuals. It could not distinguish between a strain of influenza it had encountered before and a new strain it was meeting for the first time. It could not build a targeted response to a specific pathogen and hold on to that response for future use. What life needed was a system that could look at a specific pathogen, identify its unique molecular features, build a response precisely targeted to those features, and then keep a record of that response, so that if the same pathogen appeared again, the response could be deployed instantly and with overwhelming force. What life needed, in other words, was a system that could do what we recognize in our own minds as one of the most fundamental features of experience. It needed memory. The system that evolved to provide that memory is built around two types of cells that you have almost certainly heard of, even if you are not entirely sure what they do: B cells and T cells. Both of them arise from stem cells in the bone marrow. Both of them carry on their surfaces a type of protein called a receptor, a structure shaped in a precise way that allows it to bind to a specific target. And both of them have a property that is, when you really think about it, almost unbelievable. Every B cell and T cell in your body carries a different receptor, a different shape, a different target it is capable of recognizing. Across the population of immune cells circulating in your blood and lymph tissue right now, there is an enormous diversity of these recognition tools, each one capable of binding to a different molecular target. This library of recognition tools is not built in response to the pathogens you have encountered. It is built before you ever encounter anything. The diversity is generated in advance through a molecular process that deliberately introduces random variation into the genes that code for the receptors. Your body essentially rolls the dice billions of times, creating billions of different recognition tools, in the hope that when a pathogen appears, at least some of those tools will fit. The technical name for the process that creates this diversity is VDJ recombination, a mechanism where gene segments are shuffled and joined in different combinations to produce an enormous range of different receptor sequences. The mathematics of the system are staggering. The number of different receptors that can be generated exceeds the number of different pathogens that have ever existed. Your immune system has prepared, in advance, for enemies it has never encountered and may never encounter. This is not a plan. No organism decided to build this system because it seemed like a good strategy. It evolved through billions of years of selection pressure, because organisms that happened to have more diverse receptor libraries survived more of the infections they encountered, and passed that diversity on to their descendants. The staggering preparation that exists in your immune system right now is the product of 500 million years of organisms dying from infections that better prepared organisms survived. But generating diversity is only the beginning. The library is enormous. Most of it is useless at any given moment, because most of those different receptors are not going to encounter the specific pathogen that happens to be infecting you right now. So how does the system find the right tools in the library? It lets the pathogen do the searching. When a pathogen enters the body, its molecular components circulate through the lymph nodes and the spleen, where vast numbers of B cells and T cells are waiting. As those components move through those spaces, they encounter receptor after receptor. Most receptors do not fit, but eventually, the pathogen's components find a receptor that matches a B cell or T cell whose randomly generated recognition tool happens to bind to this specific molecular target. The fit activates the cell, and it begins to divide, and divide, and divide. Within days, a single matched cell has become thousands and then millions of copies of itself, all carrying the same receptor, all capable of recognizing the same target. all armed and ready to fight the specific pathogen that triggered the response. This process is called clonal selection. The pathogen itself selects the right defenders from the library. The system does not need to know in advance which defenders will be needed. It just needs to make sure the right ones exist somewhere. Waiting. B cells, once activated, begin producing proteins called antibodies. An antibody is a molecule shaped like a Y, with two arms precisely configured to bind to the same molecular target that activated the B cell in the first place. Antibodies are released into the bloodstream in enormous quantities. They circulate everywhere, binding to pathogens when they find them, coating them, neutralizing them, marking them for destruction by other immune cells. The precision of an antibody is extraordinary. It does not just bind to bacteria in general. It binds to a specific protein on the surface of a specific type of bacterium. It is a lock shaped for one particular key. Though, like everything in biology, the fit is sometimes imperfect, and the system has ways of refining it over time. T cells do something different, and in many ways more complex. Some T cells become killers, directly destroying cells that have been infected by viruses. When a virus infects a cell, that cell displays fragments of the viral proteins on its surface. A molecular distress signal, killer T cells, whose receptors match those fragments, recognize the signal and destroy the infected cell before the virus inside it can finish replicating. The infected cell is sacrificed to prevent the infection from spreading. Other T cells become helpers. They coordinate the immune response, sending signals that activate B cells, strengthen the killer T cell response, and communicate with the innate immune system to make sure all parts of the defense are working together. And then there are the cells that matter most for the long-term story of immunity. Memory cells. When the infection is over, when the pathogen has been cleared and the battle has been won, most of the B cells and T cells that were activated during the fight die off. The army that was built to fight this specific battle is no longer needed at full strength. But a small subset of those cells, carrying the same receptors, the same ability to recognize the same target, survive. They persist for years, for decades, in some cases, potentially for a lifetime. These are memory cells, and they are the reason that if you had certain childhood illnesses, your immune system still carries the record of those encounters many decades later. The infection is long gone, but the memory lives on, in a population of cells circulating quietly in your blood and lymph tissue, waiting for the same pathogen to appear again. Stop and let that settle for a moment. Right now, in your blood, there are cells that have been waiting for years. Some of them carry the memory of infections you fought as a child, diseases you may not even remember having. Those cells have been circulating in silence for decades, maintaining a vigil against enemies that have not returned. If those enemies ever do return, those cells will recognize them in hours instead of days, and respond with a force that the original infection never faced. You will fight off the threat so fast and so completely that you may never develop symptoms at all. Your immune system contains living memories of things your conscious mind has long since forgotten. It has been keeping records, silently and without any effort on your part, of every significant battle it has ever fought. That is one of the most remarkable things about being alive. And yet, the adaptive immune system, for all its extraordinary power, has a problem built into its very foundation. If it can recognize anything, it can potentially recognize the wrong thing. The same diversity that makes it capable of responding to any pathogen also means that somewhere in the library of receptors, there are almost certainly cells that could recognize your own tissues, your own proteins, the molecular components of your own healthy cells. If those cells were activated, the immune system would turn against the body. It was built to protect. This is not a theoretical problem. It happens. And the way the immune system tries to prevent it, and the terrible things that go wrong when prevention fails, is a story that involves the most merciless quality control process in all of biology. A school that teaches its students by killing almost all of them. There is a small organ sitting in your chest, just behind your breastbone, that most people will go their entire lives without thinking about. It is called the thymus. In children, it is roughly the size of a walnut. By the time most people reach their 40s, it has largely been replaced by fat. By old age, it has almost entirely disappeared. A ghost of an organ that did its most important work before you were old enough to remember anything at all. Most people, when they learn about the thymus, are told that it is involved in the immune system, that it produces T cells, that it is important somehow, and then they move on, because nothing about that description suggests anything remarkable. But the thymus is, in a very specific and measurable sense, the most ruthless educational institution that has ever existed anywhere in the history of life on Earth. Every day, during the years when it is most active, the thymus receives a steady stream of young, immature immune cells from the bone marrow. These cells have not yet become T cells. They are precursors, blank slates, with potential but no defined identity, carrying genetic instructions that will, over the course of their time in the thymus, be used to generate the T cell receptors that define what they are capable of recognizing. The process of generating those receptors involves the random shuffling mechanism described in the previous chapter. The genes that code for the receptor are cut and rejoined in different combinations, producing a receptor with a unique shape and unique recognition capabilities. No two cells in the thymus at the same time will have exactly the same receptor. Every cell is, in this sense, an experiment, a hypothesis about what kind of recognition tool might someday be useful. The thymus then does something extraordinary. It tests each of those hypotheses, and it fails almost all of them. The first test is called positive selection. Every immature T cell must prove that its receptor can recognize the molecules that present the body's own protein fragments for inspection. These molecules, called MHC proteins, are the thymolecular identity cards that most nucleated cells in your body display on their surfaces. In complex vertebrate bodies like ours, MHC proteins carry small fragments of whatever proteins are being made inside the cell at that moment, holding them up for passing immune cells to inspect. A healthy cell displays normal fragments. An infected cell displays fragments of the pathogen living inside it. A T cell that cannot read these MHC displays at all is useless. It will never be able to understand whether a cell is healthy or in distress. So it is eliminated, quietly, without ceremony, within a day or two of failing the test. Roughly 70 to 80% of T cells fail this first test. 70 to 80% of every generation of immune cells that enters the thymus never makes it past the first checkpoint. They are discarded before they ever have a chance to do anything. The cells that pass positive selection have proven one thing: they can read the body's molecular language. Now they face the second test. And the second test is, if anything, more unforgiving than the first. The cells that have passed positive selection are now exposed to a remarkable set of conditions. The thymus contains specialized cells that express an extraordinary range of the body's own proteins. Proteins from the heart, proteins from the liver, proteins from the brain, the kidney, the pancreas, the skin. The thymus essentially recreates, in miniature, a molecular catalogue of what the body looks like from the inside. Each surviving T cell is confronted with this catalogue. Its receptor is tested against it. If the T cell's receptor binds too strongly to any of the body's own proteins, that cell is dangerous. It is a cell that, if released into the body, might attack healthy tissue. It might cause the immune system to turn against the very body it is supposed to protect. So it is killed, eliminated, before it can do any harm. This is called negative selection, and it claims the large majority of the cells that survived positive selection, when the arithmetic is complete, somewhere between 97 and 99% of the T cells that entered the thymus have been destroyed. Only 2 or 3 cells in every 100 make it through both tests and are released into the body as mature. Functional T cells, 2 or 3%. Think about what that means in practical terms. The thymus is producing and destroying immune cells by the millions every day. An enormous, continuous, metabolically expensive process of generation and elimination, all in service of finding the tiny fraction of cells that are both capable of doing the job and safe to be trusted with it. The cells that survive are the ones that hit a narrow target, sensitive enough to detect and respond to genuine threats, but not so sensitive that they react to the body's own molecules, capable of reading the MHC system, but not so reactive that they turn that capability against healthy tissue. This is the most precise biological calibration in nature, and it has to work correctly with each of the tens of millions of T cells that mature and enter the body over a lifetime. A consistent failure in either direction. Too many cells that cannot do the job, or too many that will attack the body itself, means disease or death. Most of the time, it works. Most people go through most of their lives without the calibration failing in a way that causes serious problems. But here is what the existence of this system makes inevitable. The calibration is not perfect. It cannot be perfect. The process is probabilistic, not absolute. And in the gap between nearly perfect and completely perfect, something lives that affects tens of millions of people and that medicine has been struggling to address for decades. The gap is where autoimmune disease begins. When a T cell that should have been eliminated in negative selection is not. When a cell whose receptor happens to bind too strongly to a protein in the pancreas, or the joints, or the protective sheath around nerve fibers slips through the checkpoint, what follows can be catastrophic and relentless. In lupus, the immune system produces antibodies against the body's own DNA and proteins, triggering inflammation in the kidneys, the skin, the joints, the heart, sometimes in multiple organ systems simultaneously. A person with lupus can be fighting immune-mediated damage in half a dozen places in their body at the same time. Each flare unpredictable, each shaped by factors as varied as sun exposure, stress, and hormonal changes. In multiple sclerosis, immune attack targets the myelin sheath, the fatty insulating layer that surrounds nerve fibers in the brain and spinal cord. Myelin allows electrical signals to travel quickly and efficiently. When the immune system strips it away, those signals slow or stop. The result can be vision problems, loss of coordination, muscle weakness, cognitive changes, and in severe cases, significant disability. In rheumatoid arthritis, immune activity concentrates in the lining of the joints, The inflammation that results is painful, progressive, and destructive, gradually eroding the cartilage and bone, unlike the osteoarthritis that comes with age and mechanical wear. Rheumatoid arthritis is driven entirely by the immune system, treating joint tissue as if it were an invading pathogen. In type 1 diabetes, the destruction is of the beta cells of the pancreas, the cells responsible for producing insulin. Once those cells are gone, the body has no way to regulate blood sugar without external help. The immune attack that causes it can begin years before any symptoms appear, silently eliminating the beta cell population until there are not enough left to maintain function. In each of these conditions, the immune system is doing its job. It is recognizing a target. It is mounting a response. It is fighting with everything it has. The catastrophe is not a failure of the immune system to function. It is a failure to recognize that the target it is fighting is itself. The thymus does something else worth mentioning, something that connects the precision of its selection process to a much larger story. It shrinks. Not immediately, not in childhood, when it is needed most, but after puberty. The thymus begins a slow, steady process of involution. It gradually replaces its functional tissue with fat. Its output of new T cells declines. By middle age, it is a fraction of its earlier size. By old age, it has largely ceased to function. The T cell pool in an older person is dominated by cells produced decades earlier. Cells that have been circulating for years and that carry the immune memories of a lifetime of exposures. New T cells, with fresh receptors capable of recognizing genuinely novel threats, are produced in much smaller numbers. This is one of the reasons the immune system becomes less capable with age. Not just slower or weaker in a general sense, but specifically less able to mount a strong response to new pathogens and unfamiliar threats that the existing T-cell pool has not been trained to recognize. The thymus does most of its work when you are young, produces the T-cell repertoire that will protect you for most of your life, and then quietly retires. And even with all of that, even with the involution and the declining output and the imperfect calibration that allows autoimmune disease to slip through, the adaptive immune system remains one of the most sophisticated and capable biological systems ever produced by evolution. The T cells circulating in your body right now still carry the memories of the infections you survived, still know the faces of the enemies your body has already fought, still have the ability, in most cases, to respond with speed and precision to the threats that are most likely to appear. The school is brutal. The graduation rate is 2%, but the graduates are extraordinary, and they are about to face something that neither the innate immune system nor the adaptive immune system, for all their combined sophistication, was ever fully designed to handle. Something that does not look like an invader at all. Start with a number, roughly 38 trillion. That is a current scientific estimate of the number of microbial cells living in and on your body right now. Bacteria, mostly, along with archaea, fungi, and viruses. 38 trillion individual organisms, the majority of them in your gut, existing in a relationship with your body that has been developing and refining itself for as long as your species has existed. Your immune system knows they are there. It cannot miss them. Many of these organisms carry exactly the kinds of molecular signatures that would, in any other context, trigger an immediate and aggressive immune response. They are, by the criteria the immune system uses to identify threats, indistinguishable from dangerous pathogens. And yet, your immune system does not attack them. Not because it cannot, not because it fails to detect them, but because, over the course of hundreds of millions of years of evolution, the immune system and the microbial communities that inhabit the body reached an accommodation, a working relationship, a set of boundaries, enforced by signals on both sides, that allows the microbiome to exist without being attacked, and allows the immune system to tolerate its presence without interpreting it as a threat. Human immunity is not only a war machine, it is also a peace treaty, an ongoing, constantly renegotiated peace treaty between your body and the 38 trillion organisms that live inside it. This is not a minor side note to the story of immunity. It is one of its central features. The microbiome is not simply a passenger. It is an active participant in the immune system's function. The bacteria in your gut play roles in immune development, immune calibration, and the regulation of inflammatory responses that researchers are still working to fully understand. Children raised in environments with reduced microbial diversity show higher rates of allergies and autoimmune conditions than children raised in environments with richer microbial exposure. The immune system needs to encounter microorganisms during its development in order to calibrate itself properly. Without sufficient microbial input, the calibration can go wrong. When the peace treaty breaks down, when the microbiome is severely disrupted by antibiotics, by extreme dietary changes, by illness, the consequences for immune function can be significant. The immune system loses part of the context it uses to distinguish harmless from harmful. The calibration shifts, and, sometimes, the result is a system that starts fighting the wrong enemies. The immune system is not only the art of attack, it is also the art of restraint. And restraint, it turns out, can be unlearned. Which brings us to the other side of this story. Because while the immune system is maintaining an uneasy peace with roughly 38 trillion microorganisms, it is also, in tens of millions of people, fighting a war against something it should never be fighting at all: itself. The category of disease, called autoimmune conditions, encompasses more than 80 distinct diagnoses. The exact number of people affected depends significantly on how the counting is done, and estimates vary widely. But it is clear that tens of millions of Americans are living with at least one autoimmune condition. Worldwide, the number runs into the hundreds of millions. Each of those conditions has its own specific features, its own affected tissues, its own particular pattern of damage. But underneath the differences, they share the same terrible logic. The immune system has turned against the body it was built to protect. The defenders have become the threat. The details are worth sitting with, because they make concrete what is otherwise easy to discuss in the abstract. In lupus, the immune system produces antibodies against the body's own DNA and proteins. The resulting immune complexes deposit in the kidneys, the skin, the joints, the heart, triggering inflammation in each of those tissues. A person with lupus can be managing immune-mediated damage in multiple organ systems at the same time. Each flare unpredictable, each affected by factors ranging from sun exposure to stress to infection. The disease does not announce itself clearly. It mimics other conditions. It waxes and wanes. And for many patients, the path to diagnosis takes years. In multiple scleroses, the target is myelin, the insulating sheath around nerve fibers. When the immune system strips away myelin, electrical signals in the brain and spinal cord slow or fail. The symptoms depend entirely on which nerves are affected. Vision, balance, coordination, cognition, sensation. The disease can progress steadily, or it can cycle through relapses and remissions in a pattern that is almost impossible to predict. In rheumatoid arthritis, immune activity concentrates in the joints. Inflammation erodes cartilage and bone over time, deforming the affected joints, causing pain that is not mechanical but immunological. The immune system is not responding to damage. It is causing it. In type 1 diabetes, the destruction is quiet and invisible until it is nearly complete. Immune cells destroy the beta cells of the pancreas over a period of months or years before blood sugar regulation fails and the disease announces itself. These are not infections the immune system is failing to fight. These are cases where the immune system is fighting something it should never have engaged. The war is real. The weapons are real. The damage is real. The enemy is not. Current therapies for most autoimmune diseases work by suppressing the immune system broadly, reducing its overall activity to reduce the damage it is causing. This approach works to varying degrees. The inflammation can be controlled. The progression can be slowed. Quality of life can be significantly improved. But broad immune suppression carries its own costs. A person whose immune system has been suppressed to prevent it from attacking their joints is also a person whose immune system is less capable of fighting infections. There is no current way to tell the immune system specifically to stop attacking one tissue while leaving everything else intact. This is the problem that autoimmunity reveals about the immune system as a whole. For all its specificity, for all the precision of its receptor-based recognition system, the immune system does not come with a fine-grained off switch. You cannot, at least not yet, turn off one specific immune response without affecting others. You cannot surgically correct the calibration error that is causing the damage while leaving everything else untouched. And then there is allergy. Allergy is not technically the same thing as autoimmunity. The target is different. In autoimmune disease, the immune system attacks the body's own tissues. In allergy, it attacks something external, a food protein, a pollen grain, a molecule from an animal's skin or saliva. But the underlying problem is deeply similar. A misdirected immune response causing real harm in the absence of a genuine threat. An allergic reaction is the immune system treating something harmless as if it were a dangerous pathogen. The immune system encounters peanut protein, or grass pollen, or the molecules in certain medications. It classifies them as threats. It mounts a response. In mild cases, the response produces sneezing, watery eyes, an itchy rash. In severe cases, it can produce anaphylaxis, a rapid, systemic immune activation that can cause the throat to swell shut, blood pressure to drop catastrophically, and without immediate intervention, death. The rates of both allergy and autoimmune disease have increased significantly in industrialized countries over the past several decades. This increase has happened too fast to be explained by genetic change. What has shifted is the environment. Leading hypotheses point to changes in the microbial environment as a significant contributing factor. The immune system evolved in a world full of parasites, bacteria, and other microorganisms that it learned to tolerate. When the microbial environment becomes too clean, when the developing immune system encounters too few of the organisms it co-evolved with, the calibration goes wrong. The immune system, deprived of the input it needs to learn what to tolerate, starts treating things it should leave alone as threats. This is consistent with what researchers have observed across many different populations. People in industrialized countries with high standards of sanitation have higher rates of allergies and autoimmune diseases than people in less industrialized settings, children raised on farms, with regular exposure to a diverse microbial environment, have lower rates than children raised in cities. The pattern is consistent. Even if the underlying mechanisms are still being worked out, the peace treaty with the microbiome is part of what keeps the calibration right. When that treaty holds, the immune system knows where the boundaries are. When it breaks down, the immune system can lose its bearings entirely. And in that confusion, it sometimes starts fighting wars it should never have started, against the body's own tissues, against harmless foods and environmental particles, against the very things it was built to live alongside. The war turns inward, and, from the outside, no one can see it happening. In the spring of 1796, a country doctor in rural England did something that, by the ethical standards we hold today, would be completely unacceptable. He took material from a pustule on the hand of a milkmaid named Sarah Nelms, who had contracted cowpox from a cow named Blossom. He made 2 small cuts in the arm of an 8-year-old boy named James Phipps, the son of his gardener, and he rubbed the cowpox material directly into the cuts. 6 weeks later, he deliberately exposed the boy to smallpox. By modern standards, this experiment on a child could not be conducted, approved, or defended. But in 1796, in the context of a disease that killed roughly 30% of the people it infected and left countless survivors permanently scarred or blinded, Edward Jenner's experiment became one of the turning points in the history of medicine. James Phipps did not get sick. Jenner did not understand why. He had no knowledge of B cells or T cells or antibodies or memory cells or any of the molecular machinery we have spent the last several chapters exploring. He did not know what a virus was. The germ theory of disease would not be established for another 80 years. Jenner was working entirely from observation, from the folk knowledge among milkmaids that surviving cowpox seemed to protect against smallpox, and from his own careful and methodical experiments. He called what he had discovered vaccination. From the Latin word for cow, he had no idea what he had actually discovered. What he had discovered, without the language or the science to describe it, was adaptive immunity. He had found a way to teach the body to remember an enemy before the enemy arrived. The logic of vaccination, when you understand the immune system, is almost elegant in its simplicity. The adaptive immune system builds its response through clonal selection. A pathogen enters the body. Its molecular components are encountered by the enormous library of B cells and T cells, and the cells whose receptors match those components are activated. They divide, they fight, and when the fight is over, a subset of them become memory cells, carrying the record of that encounter forward for years or decades. Vaccination exploits this process deliberately. Instead of waiting for the dangerous pathogen to appear, you introduce something that resembles it closely enough to trigger the immune response, but that cannot cause the disease itself. The immune system sees what appears to be the pathogen. It mounts a response. It builds memory cells. And then, if the real pathogen ever appears, the memory is already there, ready to deploy. The person has won a battle that never happened. They carry the immunological record of a war their body never actually had to fight. A vaccine is not a force field. It is a rehearsal, a carefully staged confrontation with something that looks enough like the threat to teach the immune system what the threat looks like, without any of the risk of the real encounter. Over the 2 centuries since Jenner made his cuts in James Phipps's arm, vaccination has been refined and expanded, almost beyond recognition. Early vaccines used live or weakened forms of the actual pathogen. Later generations used killed pathogens, or just specific protein fragments from pathogens, enough to trigger an immune response without any risk of infection. Each generation of vaccine technology represented a deeper understanding of what the immune system actually needs to see in order to build a useful memory. And then, in the early 2000s, a new approach began to emerge from research that had actually been underway for more than 3 decades. An approach that did not use the pathogen at all, not even a fragment of it. The technology was called mRNA vaccination. Every protein in your body, including the proteins on the surface of viruses that the immune system learns to recognize, is manufactured inside cells according to instructions encoded in DNA. Those instructions are first transcribed into a molecule called messenger RNA, which carries the protein-building instructions from the nucleus to the cellular machinery that assembles the protein. An mRNA vaccine delivers directly into your cells the messenger RNA instructions for building one specific protein from the target pathogen. Your cells read those instructions, build the protein, and display it. Your immune system sees the protein, which is foreign, which does not belong, and mounts a response. Memory cells are generated, and then the messenger RNA, which is inherently unstable, breaks down within days and disappears. The protein disappears with it. The immune response winds down. What remains is the memory. The vaccine is not a weakened pathogen. It is not even a fragment of a pathogen. It is a set of instructions that causes your body to briefly manufacture one small piece of what the pathogen looks like, long enough for the immune system to learn from it. And then the instructions dissolve and the piece is gone. This technology had been in development for more than 30 years before COVID-19 made it famous. Researchers had been refining mRNA approaches since the late 1980s, working through the technical challenges of delivering fragile RNA molecules into cells without triggering dangerous immune reactions before they could do their job. What COVID-19 provided was the first large-scale deployment of the technology, and the results demonstrated, in the clearest possible terms, that the approach worked. The speed with which effective vaccines were developed and deployed was not a shortcut. It was the result of decades of foundational research, reaching a moment when the investment it represented was suddenly, urgently needed. But here is what matters most about vaccination for the story we are telling. Vaccination is the first time in the history of life that an organism deliberately shaped the immunological memory it was building. Not through infection, not through natural exposure, through conscious intervention in the process of adaptive immunity. For 500 million years, immunological memory was built by disease. You encountered a pathogen. Your immune system fought it. If you survived, you had memory. The memory was real and powerful and lasting. But it came at the cost of the infection itself. Vaccination changes that equation. The memory is built without the disease. The immune system is educated by an experience that was specifically designed to be safe. The body learns the face of an enemy it has never actually fought. This is a genuinely profound shift in the relationship between an organism and its own immune system. For the first time, that relationship becomes something that can be intentionally directed. The body can be taught. The memory can be shaped. The immune system can be prepared for battles that have not yet arrived. And yet, vaccination, for all its power, works because it gives the immune system a recognizable target, a molecular face it can learn to identify. The whole approach rests on one assumption, that the enemy has a face that can be shown to the immune system in advance. A face that is foreign, a face that is clearly different from the body's own proteins. Most infectious pathogens fit that assumption reasonably well. But there is one category of threat where that assumption breaks down entirely. A threat where the face that needs to be recognized is not a foreign protein from an outside invader. A threat where the face that needs to be recognized is, in the most fundamental biological sense, your own. Vaccines are powerful tools when the enemy arrives from outside, with a face the immune system can be trained to know. Cancer is a different problem entirely, because cancer does not arrive from outside. It rises from within. It is built from your own cells, carrying your own proteins, wearing your own molecular identity. And the immune system, which has spent 500 million years learning to attack what is foreign and protect what is self, is poorly prepared by its very design to handle an enemy that looks, in almost every way, like the body itself. Teaching the immune system to remember the face of a pathogen is a problem medicine largely solved. Teaching the immune system to recognize and attack something that looks like the body itself is a different challenge entirely, one that took the entire 20th century to begin to understand, and that we are only now, in the early decades of the 21st, beginning to address in ways that actually work. In 2012, a young girl from Pennsylvania had been fighting leukemia for more than 2 years. She had been diagnosed as a small child. She had gone through chemotherapy. The leukemia had gone into remission. Then it had come back. She had gone through more chemotherapy. The leukemia had gone into remission again. Then it had come back again, worse than before. By the time she arrived at the Children's Hospital of Philadelphia in 2012, she had relapsed twice. The standard treatment options had been exhausted. Her doctors told her family that there were very few options left. Her name was Emily Whitehead. She was 7 years old. We will come back to Emily, because what happened to her is, in many ways, the culmination of everything this story has been building toward. But first, we need to understand why cancer is such a fundamentally different kind of problem for the immune system than anything it evolved to handle. Everything that has attacked the immune system throughout the history of life has had one thing in common. It came from outside. Viruses, bacteria, parasites, fungi, even the most sophisticated pathogens, the ones that have spent millions of years learning to evade immune detection, are ultimately foreign. They carry DNA that is not yours, proteins that are not yours, molecular signatures that are not yours. The immune system has been shaped by billions of years of fighting exactly those kinds of enemies. Foreign things, outside things, things that do not belong. Cancer is something the immune system was never designed to fight. A cancer cell began as one of your own cells. It carries your DNA. It displays your molecular identity on its surface through the same MHC system that every other cell in your body uses to announce itself. It uses your proteins, your signaling pathways, your metabolic machinery. When the immune system scans it, looking for the signs of foreign invasion, it has been trained to recognize. It sees something that looks, in most ways, like a neighbor. And this is not accidental. The cancer cells that survive long enough to form tumors, to grow, to spread, to threaten lives, are by definition the ones that were best at passing the immune system's inspection. The cells that looked too abnormal were detected and destroyed early, before they could establish themselves. What remains after that selection process are the cells that found the best ways to look normal. That is not a coincidence. That is evolution happening inside a single body, over weeks and months. with the immune system acting as the selecting agent. But looking like normal tissue is only the first way cancer evades the immune system. The second way is more direct, and in some respects more alarming. Cancer cells can actively suppress the immune response. The adaptive immune system, as you learned in the chapter about the thymus, has built-in mechanisms to prevent it from becoming too aggressive. Checkpoints, molecular brakes that tell T cells to stand down, to stop attacking, to accept that what they are looking at is safe and should be left alone. These brakes exist for essential reasons. Without them, the immune system would attack everything, including healthy tissue. The brakes are what prevent the immune system from destroying the body it was built to protect. Cancer has learned to use those brakes. Many cancer cells express on their surface a protein called PD-L1. This protein binds to a receptor called PD-1 on the surface of T cells. When that binding occurs, the T cell receives a signal that says, in chemical terms, stand down. What you are looking at is safe. Do not attack. This is a signal the immune system uses to protect healthy tissue from attack. It is part of the tolerance machinery that allows the immune system to coexist with the microbiome, to avoid attacking the body's own proteins, to maintain all the delicate peace treaties that keep the immune system from tearing the body apart. Cancer has hijacked that signal. It displays PD-L1 not because it is safe, but because displaying PD-L1 causes T cells to stand down. The cancer cell is waving a flag that says nothing to see here. Move along. And the immune system, because it is built to respect that flag when it appears on the surface of a cell, does exactly what the flag tells it to do. It moves along. This is not the only mechanism cancer uses to suppress immunity. Some tumors create a local environment that is actively hostile to immune cells, flooding the area around the tumor with signals that inhibit T cell function. Some cancers shed proteins that exhaust the T cells that manage to reach them, pushing them into a state of unresponsiveness where they can no longer attack even when they are in direct contact with the cancer cells. Some cancers downregulate the very MHC molecules that T cells need to see in order to recognize a cell as abnormal, essentially removing the distress signal that would identify them as targets. Cancer is a master of immune evasion, not because it was designed to be, but because every cancer cell that was not a master of immune evasion was destroyed before it could become a problem. The tumors we actually see, the ones that grow and spread and threaten lives, are the survivors of a brutal selection process in which the immune system was the selecting agent. The immune system does eliminate many cancers, probably far more than we know about, because the eliminations that succeed leave no trace. A cell becomes cancerous. The immune system recognizes it and destroys it, and the person never knows either event occurred. Immune surveillance is real and important and continuous, but immune surveillance has limits, and when a cancer cell finds a way past those limits, the game changes. The immune system, which should be the body's first and most powerful defense against cancer, becomes something closer to a bystander, and in some circumstances, worse than a bystander, because the inflammatory environment that the immune system creates around a tumor meant to fight the cancer can, in some situations, actually help the tumor grow by providing signals that promote blood vessel formation, by creating conditions that certain tumor cells can exploit. The very soldiers sent to destroy the tumor are sometimes recruited into its service. This is the most unsettling aspect of the relationship between cancer and immunity, and understanding it in detail at the molecular level took decades of careful work by thousands of scientists. But out of that work came something that Edward Jenner, working with cowpox and a small boy in rural England, could not have imagined. A new way of thinking about how to treat cancer that did not involve attacking the cancer directly. A way of thinking that went back to what the immune system already knew how to do. And asked a question that sounds simple, but that nobody had been asking in quite the right way. What if the problem was not that the immune system could not fight cancer? What if the problem was simply that it had been told not to? In the late 1980s, a researcher at the University of California, Berkeley, was spending his evenings playing harmonica in a blues band and his days asking a question that most of his colleagues considered either unanswerable or not particularly worth answering. His name was James Allison. He had grown up in Alice, Texas, a small town in the south of the state, where summers are long and brutal, and not much changes from one year to the next. His mother died of lymphoma when he was a child. His uncle died of melanoma. Another uncle died of lung cancer. By the time he was a teenager, cancer had taken 3 people from his immediate world, and he had decided, with the particular clarity that grief sometimes produces in young minds, that he was going to spend his life trying to understand it. What he was asking in those late 80s lab sessions was not, on its surface, a question about cancer at all. It was a question about T cells. Specifically, it was a question about why T cells sometimes did not respond to the things they should respond to. Why cells that were clearly capable of recognizing a target, that had the right receptor for the job, that were in the right place at the right time, simply failed to activate, failed to fight, stood down when they should have been charging forward. The answer, he eventually found, changed oncology permanently. T cells, Allison discovered, do not activate on the basis of receptor binding alone. Binding to a target is necessary but not sufficient. The T cell also needs a second signal, a molecular confirmation that says yes, this is a real threat. Proceed. Without that second signal, the receptor binding produces not activation, but something closer to the opposite, a state of unresponsiveness. This makes sense as a design feature. A system that activated T cells on the basis of receptor binding alone would be dangerous. Receptor binding can happen with harmless environmental molecules that happen to have a shape that fits a particular receptor. Building in a second signal requirement means that the immune system needs 2 independent confirmations before it commits to an attack. It reduces false positives. It protects healthy tissue. But it also creates an opportunity for exploitation. Allison was particularly interested in a protein that appeared on the surface of T cells called CTLA4. This protein, when it bound to certain molecules on the surface of other cells, delivered an inhibitory signal to the T cell, a signal that said, do not proceed, stand down. This encounter does not warrant a response. CTLA4 was a brake, one of the molecular brakes that the immune system uses to keep T cells from overactivating, from attacking healthy tissue, from causing the kind of autoimmune destruction that the thymus selection process exists to prevent. Allison's insight, developed over years of careful work, was to ask what would happen if you blocked that brake, if you prevented CTLA4 from delivering its inhibitory signal. Would the T cells that had been held in check by that signal suddenly activate? Would they start doing what they were capable of doing but had been prevented from doing? He tested the idea in mice with tumors. He created an antibody that blocked CTLA4, preventing it from signaling. He gave the antibody to tumor-bearing mice. The tumors shrank. Not in every mouse, not immediately, but in enough mice and dramatically enough that the result was unmistakable. Blocking the break released T cells that had been present in the tumor environment but had been suppressed. Those T cells, freed from inhibition, attacked the tumor. The immune system, which had been sitting in the presence of the cancer without responding to it, was suddenly fighting. The drug that eventually emerged from Allison's work is called ipilimumab. It was approved by the Food and Drug Administration in 2011 for the treatment of metastatic melanoma, a form of skin cancer that, at that stage, carried a median survival measured in months. In clinical trials, a subset of patients given ipilimumab showed responses that had not been seen before in this disease. Long-term survival, in some cases, apparent, complete response. Patients who had been told they had months to live were still alive years later. Not all patients responded. The response rates were not overwhelming, and the side effects were significant. Because blocking CTLA-4 could trigger immune responses against healthy tissue as well, producing autoimmune inflammation in the gut, the liver, the lungs, the skin. Taking the break off T cells entirely did not affect only their response to cancer. It affected their response to everything. But the proof of concept was real. The immune system could fight cancer. It had, in those patients who responded, fought cancer and won. It had simply needed the break removed to do so. At roughly the same time that Allison was pursuing CTLA-4 in Berkeley, a scientist named Tasuku Honjo at Kyoto University in Japan was studying a different protein, one he had discovered in 1992 during research on cell death. The protein was called PD-1. Honjo did not initially know what it did. He spent years working to understand its function. What he eventually discovered was that PD-1 was another break, a different break, acting through a different pathway, with different characteristics, where CTLA-4 operated primarily in the lymph nodes. At the stage where T cells are first being activated, PD-1 operated primarily in the tissues. At the stage where activated T cells had already reached their target and were deciding whether to attack, when PD-1 on the surface of a T cell binds to its partner molecule, PD-L1, on the surface of another cell, the T cell receives an inhibitory signal. Stand down. What you are looking at is safe. This is normal tissue. And as you learned in the previous chapter, many cancer cells express PD-L1 on their surfaces. They display the signal that tells T cells to stand down. They use the immune system's own tolerance machinery against itself. Honjo's work pointed toward the same therapeutic approach that Allison had taken with CTLA-4. If the problem was a brake being applied by the cancer, the solution was to block the brake. Antibodies that blocked either PD-1 on T cells or PD-L1 on cancer cells could prevent the inhibitory signal from being delivered. T cells that had been standing down in the presence of the tumor could start fighting it. The results of clinical trials with PD-1 blocking antibodies were, in some cancers, even more striking than what had been seen with ipilimumab in certain types of lung cancer, bladder cancer, kidney cancer, and lymphoma. Patients who had exhausted every other option showed responses that the field had not seen before. Some showed complete responses, with tumors disappearing entirely on imaging scans and remaining absent for years. Allison found one break. Honjo found another. Together, they changed cancer medicine. In 2018, James Allison and Tasuku Honjo were awarded the Nobel Prize in Physiology or Medicine. The citation described their discovery as establishing an entirely new principle for cancer therapy. They had not found a drug that killed cancer cells. They had found a way to release the immune system from the constraints that cancer had placed on it, and let the immune system do the killing itself. The approach that emerged from their work is called checkpoint immunotherapy. The name comes from the checkpoints, the molecular decision points where T-cell activation can be encouraged or suppressed. Allison and Honjo found the breaks at 2 of those checkpoints, and they found that removing those breaks could, in the right circumstances, transform a non-responding immune system into one capable of destroying tumors that had been growing unchecked for months or years. But checkpoint immunotherapy, for all its power, has limits that became clear as experience with it accumulated. It works by releasing T-cells from generalized inhibition. The T-cells that are released are the ones already present in the tumor environment, that already had some capacity to recognize the cancer cells, but had been suppressed. If those T-cells do not exist, or if they cannot recognize the cancer cells, even when released from inhibition, checkpoint immunotherapy does not help. And in many patients, for reasons that are still being actively researched, it does not help. Response rates vary enormously across different cancer types and different patients. In some cancers, checkpoint immunotherapy has become a standard of care that has dramatically extended survival. In others, it produces responses in only a small minority of patients. The side effects remain significant as well. Releasing T-cells from inhibition does not release them selectively, only against cancer. The same T-cells that attack the tumor can attack the gut, the liver, the skin, the lungs. The autoimmune complications of checkpoint immunotherapy can in some cases be severe, and managing them requires immunosuppressive treatment that can itself interfere with the anti-tumor response. Allison and Honjo found the breaks. They proved, beyond any reasonable doubt, that the immune system is capable of fighting cancer when it is allowed to do so. They established a principle, but releasing existing breaks can only do so much. The T-cells being released are the ones that are already there, already somewhat capable of recognizing the target, just being suppressed. What if you could do something more fundamental? What if you could take T-cells that had no particular ability to recognize a specific cancer and engineer that ability into them directly? What if you could design, from scratch, the recognition system that a T-cell would need to hunt down and destroy a particular kind of cancer cell, and then install that system into real T-cells taken from a real patient? What if you could build a living weapon designed for a specific enemy and send it back into the body that needed it? This was not a hypothetical question. By the time Allison and Honjo received their Nobel Prize, researchers at several institutions had been working on exactly this problem for more than 2 decades. And one of them had already walked into a hospital room where a 7-year-old girl named Emily Whitehead was running out of options. In April of 2012, Emily Whitehead was admitted to the Children's Hospital of Philadelphia with a fever and severely low blood counts. She was 7 years old. She had relapsed twice from acute lymphoblastic leukemia. The standard treatment options had been exhausted. Her physicians told her parents, Tom and Kari Whitehead, that without a new approach, there was very little they could do. The experimental therapy she was enrolled in had been developed over more than 2 decades by a scientist named Carl June, working at the University of Pennsylvania. It had shown responses in a small number of adult patients with similar leukemias, but it had never been tested in a child. The approach was unlike anything that had been tried before in a pediatric patient, and it carried risks that were not fully understood. The therapy was called CAR T cell therapy. The letters stand for chimeric antigen receptor T cell therapy. The name is technical. The idea, once you understand what the immune system is capable of, is almost breathtakingly logical. T cells are powerful. They can find infected or abnormal cells anywhere in the body, destroy them with precision, and remember them for years afterward. The problem, in the case of cancer, is targeting. T cells need to be able to recognize their target, and cancer cells, as you now know, are experts at avoiding recognition. What if you could solve the targeting problem directly, not by releasing existing brakes, as checkpoint immunotherapy does, but by engineering, from scratch, a recognition system that would direct T cells precisely to cancer cells, not waiting for evolution to produce the right receptor by chance, but designing it deliberately and installing it into living cells. This is what CAR-T therapy does. The process begins with the patient's own blood. A volume of blood is drawn and processed to isolate T cells. The same T cells that emerged from the thymus, that carry the memories of every infection the patient has survived, that are circulating in the blood, doing the work they were designed by 500 million years of evolution to do. These T cells are then sent to a laboratory. Scientists use a modified virus, one that has been carefully engineered to be incapable of causing disease. As a delivery vehicle, they load this virus with a gene that codes for a protein called a chimeric antigen receptor. The chimeric antigen receptor is a synthetic molecule, designed on a computer, built in a laboratory, and assembled from components borrowed from different parts of the immune system. One end of it is designed to bind to a specific protein found on the surface of cancer cells. In the case of Emily Whitehead's leukemia, the target was a protein called CD19, which appears on the surface of the leukemic B cells that were threatening her life. The other end of the receptor, once it binds to its target, delivers an activation signal directly into the T cell. The moment the receptor finds its target, the T cell activates and begins to fight. The virus carrying this gene is allowed to infect the patient's T cells. The gene integrates into their DNA. The T cells begin producing the chimeric antigen receptor on their surfaces. They have been transformed. They are still the patient's own T cells, still carrying all the memories and capabilities evolution gave them. But now they also carry something new, a synthetic recognition tool designed by human beings, pointing at a specific target on a specific type of cancer cell, a capability that 500 million years of evolution did not produce, installed in days. These engineered T cells are then grown in the laboratory over the course of roughly 2 weeks, multiplied from a small initial population into hundreds of millions of cells, all of them carrying the same synthetic receptor, all of them programmed to recognize the same target, and then they are infused back into the patient. Traditional drugs are chemicals. They enter the body, perform their function, and are metabolized and cleared. When the drug is gone, its effect ends. CAR-T cells do not work that way. When they enter the body, they are alive. They multiply further inside the patient. They move through the body, seeking their target. When they find cancer cells displaying CD19, they bind, activate, and kill, and then they move on to find the next one. Some of these engineered T cells become memory cells. They persist long after the initial infusion, circulating in the patient's blood for months or years, still carrying the synthetic receptor, still capable of recognizing and destroying any cancer cells that display the target. The therapy does not end when the infusion ends. It continues, in living form, for as long as those memory cells survive. This is what it means to call CAR T therapy a living drug. It is not a chemical that acts and is gone. It is a population of living cells that acts, multiplies, persists, and remembers. Emily Whitehead received her infusion of engineered T cells on April 25, 2012. Within days, she was critically ill. The engineered T cells, expanding rapidly inside her body and attacking the leukemic cells with a force that no previous therapy had matched, triggered an enormous release of cytokines. Her fever spiked above 104 degrees. Her blood pressure dropped. Her lungs filled with fluid. She was transferred to the intensive care unit and placed on a ventilator. For several days, the situation was so serious that her physicians were not certain she would survive. What was happening to Emily was a cytokine storm, the same phenomenon described in an earlier chapter. A massive, uncontrolled immune activation was occurring in response to the engineered T cells doing exactly what they had been designed to do. The therapy was working. It was working with such intensity that the response itself had become dangerous. Carl June and his team needed to find a way to calm the storm without destroying the therapy in the process. If they suppressed the immune system broadly, they risked killing the engineered T cells that were fighting the leukemia. They needed something more precise. Reviewing Emily's blood work, June noticed that the levels of a specific cytokine called interleukin-6 were extraordinarily high. This cytokine was a major driver of the inflammatory storm, and there happened to exist, already approved for the treatment of rheumatoid arthritis, a drug that specifically blocked interleukin-6. It had never been used for this purpose before. There was no established protocol. There was no clinical experience with it in this context. But the situation was desperate, and the reasoning was sound. Emily received the drug. Within hours, her condition began to improve. The cytokine storm subsided. Her fever came down. Her blood pressure stabilized. Over the following days, she came off the ventilator. She was moved out of the intensive care unit. On May 3, 2012, Emily Whitehead's 7th birthday. Her doctors examined her bone marrow for signs of leukemia. There was none. Complete remission. Emily Whitehead is alive today. More than a decade after that birthday, she has remained cancer-free. Her blood still carries, as far as anyone has been able to measure, engineered T cells still circulating, still ready. Some long-term responders to CAR T therapy may be functionally cured, though physicians remain careful with that word, because CAR T is still a young field, and not every patient's story ends the way Emily's did. Her story became the public face of CAR T therapy, the image of a small girl given a last chance on an experimental treatment, who nearly died from the treatment itself, who woke up on her birthday without leukemia, resonated in a way that scientific papers rarely do. The Food and Drug Administration approved the first CAR T therapy in 2017. Several more followed. As of the mid-2020s, multiple CAR T therapies have been approved for a range of blood cancers, including certain leukemias, lymphomas, and multiple myeloma. The results in some of these cancers have been extraordinary. Patients who had exhausted every other option, patients for whom medicine had nothing left to offer, have achieved complete and durable responses. But it is important to be precise about what CAR T therapy has achieved and what it has not yet achieved. It works best in blood cancers, where cancer cells are circulating in the blood and lymph system and are therefore accessible to the engineered T cells. in solid tumors, the cancers that form masses in the lung, the breast, the colon, the pancreas. The approach faces serious challenges that have not yet been fully solved. The interior of a solid tumor is a hostile environment for immune cells. Immune suppressive signals are concentrated there. The physical structure of the tumor can prevent T cells from penetrating effectively. Finding targets that are present on cancer cells but absent on healthy tissue is harder in solid tumors than in blood cancers. The side effects remain significant. Cytokine release syndrome, the storm that nearly killed Emily, is a known risk that has been better characterized and managed since her treatment, but that still occurs and still requires careful monitoring and intervention. A condition affecting the nervous system, which can cause confusion, seizures, and in rare cases, more serious neurological complications, is another potential complication, and the cost is extraordinary. CAR T therapies carry price tags that can reach hundreds of thousands of dollars per treatment. The manufacturing process is complex, time-consuming, and requires specialized facilities. For each patient, a completely individualized product must be created from that patient's own cells. Access to these therapies, even in wealthy countries, is not universal. These are real limitations. They are where the next generation of research is focused. Researchers are working on ways to manufacture CAR T cells more quickly and cheaply, exploring the use of T cells from healthy donors rather than from patients themselves, developing new strategies for getting engineered T cells into solid tumors and keeping them active there, and beginning to apply the same engineering principles to conditions beyond cancer, including autoimmune diseases, where the goal would be not to destroy cells, but to silence the specific immune activity causing harm. The T cell that emerged from Emily Whitehead's blood in 2012, that was taken to a laboratory and given a new receptor and multiplied into an army and sent back into her body to find and destroy the leukemia that standard medicine could not, carries within it the logic of everything that came before. 500 million years of adaptive immunity. The receptor-based recognition system that evolution built in ancient fish, the memory cells that outlast the infections that created them, the thymic selection process that teaches T cells the difference between self and not self. All of that ancient biology, preserved and refined across hundreds of millions of years, was present in Emily's T cells before anyone touched them. What the researchers added was one thing: a new receptor, a recognition tool that nature did not produce, pointing at a target that evolution did not prepare for, installed into cells that a billion years of biological history had made extraordinarily capable of doing exactly what was needed. The ancient system, given a new set of instructions, and it saved a child's life. Go back to the ocean. Not the ocean as you know it, with its coastlines and its abundance of visible life. Go back to the ocean as it was 3.5 billion years ago. Dark, varm, almost entirely silent. A world where the most complex things in existence were single cells drifting in the dark, dividing, surviving, doing nothing that any observer could have recognized as purposeful or directed. In that ocean, a bacterium was infected by a virus. We do not know which bacterium. We do not know which virus. We do not know exactly when or where in that vast, dark water the encounter took place. But we know that encounters like it were happening constantly, in every corner of that early ocean, because the pressure between cells and the things that invade them is as old as life itself. We also know something else. In some of those encounters, the bacterium survived, and in surviving, it left behind a molecular trace of the encounter, a small piece of the invader's genetic code tucked away in its own DNA, a record of the fight that changed how it would respond if the same enemy ever appeared again. That trace was the beginning. Not the beginning of immunity in the full sense we have explored across these chapters, not the beginning of T cells or antibodies or the astonishing architecture of adaptive immunity, but the beginning of the idea underneath all of it, the idea that an encounter with a threat can leave something behind. That survival is not just about winning the fight in front of you, but about carrying forward something that makes the next fight different. Everything in this story, everything in the 4 billion years between that ancient ocean and the moment a 7-year-old girl in Philadelphia woke up on her birthday without leukemia, is the story of what happened to that idea, how it grew, how it became more sophisticated, more targeted, more powerful, how it was built into the architecture of every complex animal that has ever lived, how it failed and was worked around, and failed again, and was worked around again, how it was eventually studied, understood, and for the first time in the history of life, deliberately extended by the very organisms it was built to protect. That is the arc of the story. But it is not the end of the story, because there is no end. The war does not stop. This is the part that is easy to lose sight of in the excitement of what medicine has achieved. CAR T therapy is real. Checkpoint immunotherapy is real. mRNA vaccines are real. The advances of the past few decades represent genuine and extraordinary progress in our ability to understand and work with the immune system. People are alive today who would not be alive without these tools. Emily Whitehead is alive. Thousands of others are alive. The progress is real, and it matters enormously. But the war continues. Viruses mutate. They are mutating right now. In patients, in hospitals, and in animals, in forests, and in humans going about their daily lives, generating variation at a speed that still exceeds almost anything else in biology. Most mutations are neutral or harmful to the virus. Some are not. Some give the virus new capabilities, new ways to evade immune responses that had previously been effective, new ways to spread and cause damage. Bacteria develop resistance to antibiotics, to immune mechanisms, to the conditions that would previously have killed them. The same selective pressure that shaped the immune system over billions of years is shaping bacterial populations right now, in real time, in response to every antibiotic we use and every immune mechanism that targets them. The bacteria that survive are the ones that found a way around the thing that was killing the others. Their descendants carry that capability forward. Cancer evolves inside individual patients, in real time, under the selective pressure of treatment. The leukemia cells that survive CAR T therapy are the ones that found a way to avoid the engineered T cells. One of the primary mechanisms by which CAR T therapy fails in some patients is the loss of the target protein, CD19, or whatever other molecule the CAR was designed to recognize. The cancer cells stop displaying it. The engineered T cells, looking for a target that is no longer there, cannot find anything to fight. The cancer, now wearing a different face, can grow back. This is not a failure of the therapy. It is evolution. It is the same process that has been operating since the first virus infected the first cell in the first ocean. Apply a pressure to a population of replicating entities, and the ones that can survive that pressure will. The ones that cannot, will not. There is no way to win that game permanently. There is no treatment so complete that it eliminates every last cancer cell and leaves no possibility of recurrence. There is no vaccine so perfect that a sufficiently mutated pathogen cannot eventually get around it. There is no antibiotic so powerful that bacteria will never develop resistance to it. These are not failures of medicine or of the immune system. They are consequences of the nature of life itself. Living things replicate. Replication produces variation. Selection acts on variation. This is the engine that produced the immune system. It is also the engine the immune system is always fighting against. You cannot turn off that engine without turning off life. So the question that matters is not whether the war ends. The question is what it looks like to be in that war with the tools and knowledge we have now, compared to what it looked like before. And there, the picture is genuinely and powerfully different. For most of human history, the immune system fought alone. It used the tools that evolution had given it over 500 million years of adaptation. And humans had no ability to meaningfully assist or direct that process. When the immune system worked, people recovered. When it did not, people died. There was essentially nothing medicine could do to change which outcome occurred. The germ theory of disease, established in the latter half of the 19th century, changed the intellectual framework without immediately changing outcomes. Knowing that bacteria cause disease was enormously important. It led to antisepsis, to the sterilization of surgical tools, to the understanding that contaminated water was killing people by the thousands. These were real advances, and they saved enormous numbers of lives. But they were primarily about prevention, about keeping pathogens away from people, rather than about helping the immune system fight them once they arrived. Antibiotics, developed in the 20th century, were a genuine turning point. For the first time, medicine had tools that could directly kill certain pathogens inside the body, supporting the immune system's work, or doing what the immune system alone could not do quickly enough. The reduction in mortality from bacterial infections over the 20th century is one of the most significant changes in human health in history. Vaccines extended this further, allowing the immune system to prepare for specific pathogens before exposure. The eradication of smallpox, one of the deadliest diseases in human history, was accomplished entirely through vaccination, through the deliberate manipulation of immunological memory at a global scale. And now, in the early decades of the 21st century, medicine has developed the ability to reach inside the immune system itself and modify it at the cellular and molecular level, to engineer T cells, to block immune checkpoints, to deliver synthetic messenger RNA that instructs the body to build its own immune targets, to design therapies that are not chemicals but living cells, that persist and adapt and respond to conditions inside the body in ways that no conventional drug can. This is a fundamentally different kind of medicine than anything that came before it. Not just more effective, different in kind. Medicine that works with the immune system at the level of its own mechanisms, that understands those mechanisms well enough to modify them deliberately, that treats the immune system not as a fixed given, but as a system that can be extended. Researchers are working on allogeneic CAR T therapy, sometimes called off-the-shelf CAR T, using T cells from healthy donors that have been modified to avoid triggering rejection in recipients. The goal is a product that could be manufactured in advance, stored, and given to any patient who needs it, at a fraction of the current cost and in a fraction of the current time. Personalized cancer vaccines are in clinical trials, designed not to prevent cancer, but to teach an individual patient's immune system to recognize the specific mutations present in that patient's specific tumor. Engineered regulatory T cells are being explored as a potential treatment for autoimmune diseases, using the same technology that makes CAR-T therapy possible, but directing it towards silencing specific immune responses rather than amplifying them. The thymus itself is a target of research. Its involution with age, its declining output of new T cells, is one of the factors that makes the aging immune system less capable of responding to novel threats. Early work in animal models has shown that it may be possible to partially restore thymic function, to give the aging immune system some of the generative capacity it had in youth. Whether that translates into meaningful benefits in humans, and whether the benefits outweigh the risks, is an active area of investigation. These are not guarantees. Not every approach in development will succeed. Not every patient who receives CAR-T therapy today will respond the way Emily Whitehead responded. Not every cancer will yield to checkpoint immunotherapy. Not every autoimmune disease will be treatable by engineering regulatory T cells. The history of medicine is full of promising approaches that turned out to be harder than they looked, and there is no reason to expect that the next generation of immune therapies will be exempt from that pattern. But the direction is clear. The movement of knowledge is in one direction, toward more understanding, toward more precise and targeted intervention, toward a relationship between medicine and the immune system that becomes, with each generation of research, more sophisticated and more aligned with what the immune system actually is and how it actually works. Now come back, one final time, to the bacterium in the ancient ocean, 3.5 billion years ago, infected by a virus. The encounter is random. The outcome depends on chemistry and chance. If the bacterium survives, it may leave behind a molecular trace of the encounter. That trace is not intelligence. It is not purpose. It is not anything the bacterium chose or designed or experienced in any meaningful sense. It is simply chemistry, producing an outcome that happens to be useful. But that outcome is the seed of everything. From that trace, over billions of years, through the blind pressure of selection acting on variation, life built the restriction enzyme, and the CRISPR array, and the macrophage, and the cytokine, and the antibody, and the T cell, and the thymus, and the memory cell, and the checkpoint, and the engineered receptor. It built all of it without a plan, without a goal, without any awareness of what it was building or why. And then, at the end of that 4 billion year process, something happened that had never happened before. One of the products of that process began to understand the process itself. A scientist in Texas grew up watching cancer take the people he loved, and spent 30 years asking why the immune system was not fighting it, and found an answer that changed what was possible. A scientist in Japan followed a different thread through a different laboratory on a different continent and found another part of the same answer. Researchers in Pennsylvania took the cells of a dying child, gave those cells a new set of instructions designed by human minds, and sent them back into her body to finish a fight that 500 million years of evolution had prepared them for, but not fully equipped them to win. The war did not end in that hospital room in Philadelphia. It has not ended. It will not end. But something shifted, something that cannot be unshifted. For 4 billion years, the immune system fought on terms set entirely by the blind pressure of natural selection, adapting through the deaths of the organisms that could not adapt. No guidance, no direction, no understanding of the war it was fighting. That has changed. The immune system still fights. It always will. But now, for the first time in 4 billion years, it fights alongside something that can see the battlefield. that can read the mechanisms, that can, in at least some cases, rewrite the instructions. The war that began in an ancient ocean, between a bacterium and a virus that neither of them knew was happening, is the same war that is happening in every human body alive on Earth right now. The same logic, the same principles, the same relentless pressure between the things that invade and the systems built to stop them. But one side of that war has learned something that no side in any war in 4 billion years of life has ever known before. It has learned to think. And it is only just beginning to understand what that means.